a postdoctoral associate work at Cornell University. He has been associate professor at Iwate University and presently he is professor at Tohoku University. His research involves studies on gene expression during reproduction in plants. Please welcome Professor Toriyama.
Thank you. It's my great pleasure to have an opportunity to introduce our study on cytoplasmic male sterility, CMS. I am Kinya Toriyama, and Torikin is my international nickname, so you can call me Torikin. This slide shows you the background. Hybrid rice has been planted more than 30% of wild total cultivated area. I don't know the situation in Thailand, but many countries have succeeded with hybrid rice. And about 95% of the hybrids are based on a single cytoplasm, CMS. So to avoid the potential threat of vulnerability of hybrid rice, development of several CMS restorer systems is desirable. So the alloplasmic CMS is considered to be caused by incompatibility between nucleus and mitochondria. When we combine the nucleus of cultivar and mitochondria of distantly related species or wild rice, we can make a substituted line, and then the nucleus and mitochondria fight each other, so battle like this. So this cell looks like angry face, doesn't it? So there is a battle here. In these mitochondria, there is an unusual ORF which produces uncomfortable peptide or a toxic peptide that results in pollen to die, resulting in male sterility. But when there is a fertility restorer gene in the nucleus, it reduces the production of such toxic peptide and then resulting in fertility restoration. So this is the examples of CMS in rice. CMS is derived from Oryza and most widely used for hybrid rice breeding, as I told you before. So more than 90% of hybrid rice rely on this cytoplasm that account for 1/10 of wild rice area. And CMS is derived from Chinsaboro2 and restorer G is RF1. CMS is derived from red rice and restorer G is RF2. CMS is derived from Oryza and the restorer G is RF17. And these are the pollen grains at the mature flowering stage. And in case of CMS, the pollen is almost empty. In case of BT, the pollen starch accumulation is less abundant than normal type. And in case of LD, the starch accumulation is more abundant than BT, but still less than normal type. In case of CW, the pollen looks like normal, but it lacks germination ability. So these are the isogenic lines, so we can say the mitochondria determine the fatal pollen grains. Let me start with CMS. BT mitochondria contain the ORS79 downstream of ATP6 and they are co-transcribed. And ORS79 produce a toxic peptide resulting pollen abortion. And RF1 for CMS encodes PPR protein, that is the pentatoricopeptide repeat protein. And it's a RNA binding protein and transported to mitochondria. And then it binds to ATP6 or 79 transcripts and then promotes processing here and resulting in no translation of ORF79 and then resulting in fertility restoration. So other than CMS, there is a couple of Oryza wild rice carrying CMS cytoplasm. And these wild rice strains are known to cause gametophytic CMS and fertility restoration. We investigated ORS79 and RF1 alleles in these wild rice, and found there is the ORF79 present and also some sequence variants. And RF1 protein encoded by these alleles contains some amino acid substitutions. And in case of W1109, it doesn't have ORF79, and this is a different type. So we can say different RF alleles will be interact with respective mitochondrial gene in a gene for gene fashion, like a pathogen virulence gene and plant disease resistant genes. So today I want to talk about a little bit about more on non-ORF79 type CMS. So RT98 CMS and RT102 CMS and fertility restore line have been obtained by successive backcrosses with W1109 and W1125 and Oryza sativa Tytune 65. And in case of RT98 CMS line, the pollen grain looks normal, but it lacks germination ability. In case of RT102, it has two types of pollen grains. One is almost empty, and the other is spherical and darkly stained with I2KI, but still, they both types lacks germination ability. We searched for candidates of CMS-associated genes by this strategy. At first, we determined the mitochondrial whole genomic sequence using GXFLX and look for novel ORF, which is absent in the standard cultivar Nippon Valley. And then we look for ORF that is chimeric to non-mitochondrial gene or that encodes the transmembrane domain. And finally, we examine the expression pattern, which will be different in the presence of RF1. We took this strategy because many CMS-associated genes also in other plants have the similar characteristics. So this is one example in BT-CMS ORF79. It is chimeric to COX-1, as I show you in blue color here. And TM-HMM analysis indicated one transmembrane domain here. And transcript size is different in the presence of RF1 or absence of RF1. So that's why we took this strategy. And this is the case of RT98. We determined the whole mitochondrial genome, and then we found that 27 ORF absent in Nippon Valley, and 6 ORF, as shown here, show a chimeric structure or encode the transmembrane domains. So this is one example, ORF113, it shows the transmembrane domains here. And then we carried out northern blot analysis using these ORF as probes. And this is a northern blot analysis of mitochondrial RNA. And A is RT98 CMS line. A in red is a CMS line. And C in blue color shows RT98 restorer line with larger left. So, and T is the Tytune 65. And we only found the transcript difference when we probe with ORF113, as I show you here. And so, A in red color, the CMS line shows a 2.2 kb band and 1.1 kb band here. But in case of C in blue color, it shows a 2.1, 1.4, and 1.0 kb. So the processing of RNA is different in the presence of RF1 or not. So this is the genomic structure around ORF113. And there is ATP4 and COX3 downstream of ORF113. And this is northern blot analysis of mitochondrial RNA, and ORF113 probe, ATP4 probe, and COX3 probe all show the similar expression patterns. And this is RT-PCR of answer total RNA using the primers here and here. And it amplifies 2.5 kb band, the same size of this band. So we conclude ORF113 is co-transcribed with ATP4 and COX3, and the transcripts are processed distinctly by RF factors. So, next we take a close look at the flanking sequence of ORF113 and found minus 151 to plus 11, as shown in red color here, is completely identical to that of NAT9. So the ORF113 shows chimeric structure to NAT9. So the ORF113 has a set of features characteristic to CMS-associated candidate genes. So next we investigated RT102, and similarly we found 27 ORF absent in Nippon Valley, and 6 ORF as shown here show a chimeric structure or encode the transmembrane domains. And this is the examples of ORF352. It has 3 transmembrane domains here. And then we carried out Northern blot analysis using these ORF as probes, and only ORF352 shows the different transcript sites depending on the RF gene. So the ORF352 includes the 5' site of ORF284 of Nippon Valley and unknown origin, and this part is similar to ORF224 of Nippon Valley, and this part is similar to 3' side of ORF288 of Nippon Valley. And this is again the Northern blot analysis of mitochondrial RNA, and A is a CMS line with small RF, and C is a restorer line with large RF. As you can see here, in A CMS line, in A line here, you can see a 2.7 kb band, but it disappeared in C line, but new smaller bands appeared here. And this is RT-PCR of mitochondrial RNA using these primers, using these as primers. And the primers shown in red color gives the band in A line here, but not in C line. So it indicates cleavage of RNA will take place around here. So this is genomic structure around ORS352. ORS352 exists between RPL5 and RPS3, and this is Northern blot analysis again, probed with RPL5 and ORS352. And in case of CMS line, A in red color shows a 2.7 kb band, but smaller bands in the C line. And this is RT-PCR using this and this primers. It amplifies the bands here in A line, CMS line, but not in the restorer line. So we conclude ORF352 is co-transcribed with RPL5, and the transcripts are processed by RF factors. So this is the summary of CMS-associated gene and RF genes. We suppose the RF gene for RT102 and RT98 is involved in processing of ORF352 and ORF113. So it may be a PPR gene, and we are now doing the fine mapping. So I hope I will tell you the sequence in the next or the next meeting. One more thing I want to say from this slide is restoration of fertility by IR24. IR24 is effective for most of the fertility restoration, but not effective for fertility restoration for CW type CMS. This is derived from Oryza W1 strain. So I want to emphasize the possible merit of CW CMS and RF17. RF17, a restorer gene of CW CMS, has been found only in the CWR line, but not in other cultivars or other restorer lines. So we suppose any commercial indica cultivars will act as maintenance lines for CW CMS. This will compensate for demerits of WHCMS. As you may know, the most indica cultivars possess RF genes for WHCMS and cannot be used as maintenance lines. But CWCMS will provide CMS line of indica cultivars. So I want to have a comment on this point from the point of view of hybrid rice breeding. So this study is supported by Program for Promotion of Basic and Applied Research for Innovation in Bio-Oriented Industry. And this is a mascot character, Okome-chan, of our laboratory. It represents rice grain and DNA double helix. He is running for the future. Thank you for your attention.
Thank you very much for very interesting results about male sterility in rice, which is an important component of hybrid seed production. Friends, if you have any questions or comments to Professor Toriyama. Yes, Professor Dabe.
Question: did you check presence of the ORF113 in the current hybrid rice production system? Current master line used in hybrid rice production. Current used.
His question is ORF352 and ORF113 is present in the current hybrid rice, but we haven't checked. But we know the WSCMS has a similar sequence to ORF352, but we haven't checked it yet. Thank you.
All right. There are no more questions. Thank you, Professor.
Thank you.
Our next speaker